Hello, welcome to Biology, the scientific study of life, the molecular basis of inheritance, life's operating instructions. In April 1953, James Weston and Francis Crick shocked the scientific world with an elegant double helical model for the structure of the deoxyribonucleic acid, or DNA. Over the past 50 years, their model, Francis Crick and Weston, James Weston, their model has evolved from a novel proposition to an icon of modern biology. DNA is the substance of inheritance, is the most celebrated molecule of our time. Mendel's heritable factors and Morgan's genes on chromosomes are in fact composed of DNA. Chemically speaking, your genetic endowment is the DNA contained in the 46 chromosomes you inherited from your parents. Of all nature's molecules, nucleic acids are unique in their ability to direct their own replication from monomers. Indeed, the resemblance of offspring to their parents has its basis in the precise replication of DNA and its transmission from one generation to the next. Hereditary information is encoded in the chemical language of DNA and reproduced in all the cells of your body. It is this DNA program that directs the development of your biochemical, anatomical, physiological, and to some extent behavioral traits. In this chapter we will learn how biologists deduced that DNA is the genetic material, how Weston and Crick discovered its structure, and how cells replicate and repair the DNA, the molecular basis of inheritance. DNA is the genetic material. Today, even school children have heard of DNA, and scientists routinely manipulate DNA in the laboratory and use it to change the heritable characteristics of cells. Early in the 20th century, however, the identification of the molecules of inheritance loomed as a major challenge to biologists. The search of the genetic material scientific inquiry, once T.H. Morgan's group showed that genes are located on the chromosomes, the two chemical components of chromosomes, DNA and protein, became the candidates for the genetic material until the 1940s. The case of proteins seemed stronger, especially since biochemists had identified them as a class of macromolecules with great heterogeneity and specificity of function, essential requirement for the hereditary material. Moreover, little was known about nucleic acids, whose physical and chemical properties seemed too uniform to account for the multitude of specific inherited traits exhibited by every organisms. Okay, this view gradually changed as experiments with macroorganisms yielded expected results. As with the work of Mendel and Morgan, a key factor in determining the identity of the genetic material was the choice of appropriate experimental organisms. The role of DNA is heredity was first worked out by studying bacteria and the viruses that infect them, which are far simpler than pea plants, fruit flies, or humans. In this section, we will trace the search for the genetic material in some detail as a case study in scientific inquiry. Evidence that DNA can transmission bacteria. We can trace the discovery of the genetic role of DNA back to 1928. Frederick Griffith, a British medical officer, was studying Streptococcus pneumonia, a bacterium that causes pneumonia in mammals. Griffith had two strains, varieties of the bacterium, a pathogenic disease-causing one and non-pathogenic harmless strain. He was surprised to find that when he killed the pathogenic bacteria with heat and then mixed the cell remains with living bacteria of the non-pathogenic strain, some of the living cells became pathogenic. Furthermore, this new trait of pathogenicity was inherited by all the descendants of the transformed bacteria. Clearly, some chemical components of the dead pathogenic cells caused this heritable change, although the identity of the substance was not known. Griffith called it the phenomenon transformation. Now defined as a change in genotype and phenotype due to the assimilation of external DNA by a cell. This use of the word transmission should not be confused with the conversion of a normal animal cell to a cancerous one. 40 years Griffith work set the stage for a 14 years search for the identity of the transforming substance by American bacteriologist Oswald Avery. Purified various types of molecules from the heat-killed pathogenic bacteria, then tried to transform it live a non-pathogenic bacteria with each type. Only DNA worked finally in 1944. Avery and his colleagues Macklin, McCarthy, and Colin McLeod announced that the transformed considered considerable skepticism in part because the lingering belief that proteins were better candidates for the genetic material. Moreover, many biologists were not convinced that the genes of bacteria would be similar in composition and function to those of more complex organisms, but the major reason for the continued doubt was that so little was known about DNA. Notice that inquiry: can the genetic trait of pathogenicity be transferred between bacteria? Experiment. Bacteria of the S-smooth strain of Streptococcus pneumonia are pathogenic because they have a capsule that protects them from an animal's defense system. Bacteria of the R-rough strain lack a capsule and are non-pathogenic. Frederick Griffith injected mice with the two strains as shown below. Living cells, the first one, notice that. The first one, living S cells control, and notice the result also. Notice the result, mouse dies. And next one, living R cells control. Notice the result also, mouse healthy. Heat killed S cells control. Notice the result also, mouse healthy. Mixture of heat killed S cells and living R cells. The result also, mouse dies. Living, notice also, living S cells are found in blood sample. Conclusion. Griffith concluded that the living R bacteria had been transformed into pathogenic S bacteria by an unknown heritable substance from the dead cells. Okay, thanks for